All right, I want to show you guys this inside Scientific America about the coronavirus. So we looked at this before in lectures 1 and 2, and I want to show you a little bit more now that we're further into the immune system. So, how the immune system responds. Infected cells send out alarms to the immune system to try to neutralize or destroy the pathogens. But the viruses can prevent or intercept the signals, buying time to replicate widely before a person shows symptoms. When infection begins, the innate immune system tries to immediately protect lung cells. The adaptive immune system gears up for a greater response. Here's an infected cell. Here's some interferons and a macrophage coming to the site. An infected cell releases interferon proteins that alert neighboring cells to create molecules that try to stop virus particles from entering or reproducing. The red dots are the virus. Interferon also beckons cells such as macrophages in the bloodstream that can engulf virus particles. This happens within 0 to 3 days. The adaptive immune system follows. Interferon also alerts B cells. They produce neutralizing antibodies that might recognize parts of the spike protein and bind to it, preventing the spike from grabbing on to a lung cell. Interferon also recruit T cells, which can destroy viruses and kill infected cells before viruses inside them burst out. Some B and T cells become memory cells that can quickly identify and fight a future invasion by the virus. This is in days 6 to 11. The virus takes countermeasures. SARS-CoV-2 uses several tactics to thwart the immune system's response. Tactic 1. The virus spike may camouflage itself with sugar molecules. They flex and swing, potentially blocking antibodies from attaching to the virus, neutralizing it. Normally, sensor proteins recognize incoming viruses as foreign and tell the cell nucleus to turn on genes for making messenger RNA molecules. The molecules deliver instructions to the ribosome to make the interferon proteins that exit the cell to alert the immune system cells. Tactic 2. Several SARS-CoV-2 proteins are thought to block sensor proteins from acting or to interfere with instructions to the ribosome. Drug and vaccine interventions. Commercial and university labs are investigating well over 100 drugs to fight COVID-19, the disease the SARS-CoV-2 virus causes. Most drugs would not destroy the virus directly, but would interfere with it enough to allow the body's immune system to clear the infection. Antiviral drugs generally stop a virus from attaching to a lung cell, preventing a virus from reproducing if it does invade a cell, or dampen an overreaction by the immune system, which can cause severe symptoms in infected people. Vaccines prepare the immune system to quickly and effectively fight a future infection. Drug target 1. Prevent the virus from entering the cell. A drug or therapeutic antibodies could lock onto the spike protein, preventing it from binding to a lung cell's ACE2 receptor. A drug could also attach to a protease enzyme and prevent it from cutting the spike protein so the virus cannot fuse with the cell. Drug target 2. Encouraging defective viruses. A drug could interfere with the viral RNA polymerase enzyme, which works with another enzyme called exon, not shown, to fix mistakes in copied viruses that would disable those viruses, leading to more bad copies and fewer good ones. Drug target 3. Shut down the virus. A drug could interfere with the lung cell proteins the virus needs, such as those involved in making virus proteins or in making the vesicles the virus uses to copy its genome. Drug target 4. Reduce hyperimmune response. Immune cells can destroy too many lung cells, creating enough mucus-like waste to suffocate the lungs, forcing victims onto ventilators. Overproduction of an alarm protein or cytokine such as interleukin-6 can put immune cells into overdrive. Drugs could inhibit some of the cytokines by binding to them. Vaccine options. A vaccine exposes the immune system to a safe version of a virus so it can practice making antibodies that will stop the pathogen and commit the exercise to memory so it is ready to fight the real virus during an infection. Vaccine makers are pursuing a variety of strategies for formulating and mass producing vaccines, or they were. now they have them. How vaccines work. Antibody preparation. The vaccine version of a SARS-CoV-2 virus presents various molecules called antigens that belong to the real virus. Antigen-presenting cells grab them and provide them to helper T cells and B cells. The T cells help B cells turn on to produce antibodies that could bind to the actual virus. The helper T cells also tell killer T cells to devise ways to destroy lung cells that are infected. Some of the B and helper T cells turn into memory cells that store the instructions so they can quickly spark B and T cells into action during an infection. SARS-CoV-2 vaccine development strategies. Experts are exploring at least 6 strategies for making vaccine versions of the virus. 3 of them involve injecting a modified version of the virus into people, like a weakened virus, an activated virus, or pieces of a virus. 3 involve mapping genes from the virus, such as those for the spike protein, inserting the blueprints into DNA, RNA, or a safe virus and injecting those into people. DNA, RNA, safe virus, ribosomes, and host cell make virus pieces, and the cell prevents them to T cells, triggering an immune response.